So this is part 2 of the hybrid capture lecture. So this is what the cervical histology looks like. So histology is taking a biopsy through the surface of the cervix right through into the underlying connective tissue where the blood vessels are. So up here is the epithelium, down here is the connective tissue, there is the basement membrane, you can't see it, but sat on it is a layer of basal cells. These are proliferative cells. These up here are non-proliferative cells which are maturing, aging, and then the top cells will die and detach from the tissue and then be replaced by more cells. So as we progress from a low-grade intraepithelial neoplasia to a high-grade, you can see here in the low-grade you've got highly proliferative cells in sort of the bottom third. Some of them punching their way up to the top. Here's a little pocket of them there. But then the cells in this region here look fairly normal. So if you do the cervical smear, you'd scuff off some of these cells, maybe some of these, and get a lesion that looks low grade like this. A high grade lesion has got the proliferative cells from bottom to top. It's not yet invaded through the basement membrane. So when you do a cervical smear, you get cells that look like these, which basically will look like cells down at the bottom here, highly proliferative. Now these cells shouldn't be at the top, but they are here. In fully fledged cancer, so invasive cancer, a squamous cell carcinoma, you can see you've got invasive cells that have invaded through the basement membrane and can access the blood supply. So this is, by definition, invasive cancer. This is a CIN grade 3. So that's what is currently done. You do a cervical smear, you look at the morphology of the cells. If the cells look abnormal on cytology, perform a biopsy, and then that will tell you whether there's invasive cancer or just a CIN lesion. Now the problem at the moment is lots of people are biopsied or have laser ablation for lesions which are very low risk in terms of cancer. So molecular methods are aimed at detecting whether high risk virus is there and then only treating those patients with high risk virus types which are likely to go on for, possible to go on for cancer, only treat those by some of those invasive techniques. So we have lots of molecular methods that can detect HPV. Now basic PCR will easily do it, but just detecting HPV is not the issue. We can detect HPV in a common verruca. That's not going to cause cancer. So if you only detect HPV, then we get a huge number of false positives and not going to go on and form cancer. Oversensitivity is a key issue here. A very small lesion that's not going to really do anything in the near future is not particularly a concern. A great big lesion is. So what we need to do is discriminate between high and low risk types, and these viruses differ between a few base pairs in key genes. So we can define type 16 by one key single base pair change. So if we can find the test that differentiates between type 16 and everything else, then we know a patient has got type 16. So any assay must discriminate between low risk types and high risk types. So basic PCR, as I mentioned, is not great because it just detects HPV or no HPV. Allele-specific PCR is suitable because it can detect the difference between one high-risk type and the reference sequence, but you have to perform lots of PCRs to identify each of the couple of dozen high-risk types. So it is used. There are allele-specific PCR kits that are routinely used in NHS diagnostics labs, but as for reasons that I've already discussed, like the unidirectional workflow requirement for PCR-based technology and the generation of lots of PCR products that could contaminate future experiments, PCRs are not favoured in a cytopathology lab because the cytopathology staff there are not experienced molecular biologists. So they favoured this technique called hybrid capture, which doesn't generate any PCR product amplification. In situ hybridization, by the way, is a way of detecting HPV RNA using labelled RNA probes on cells and sections. It's not really used. So we'll focus on hybrid capture, where we're going to use an RNA probe to bind and detect known variants of the HPV genome and detect HPV DNA. So the hybrid capture technology comes from the fact that we are using a DNA-RNA hybrid to detect HPV DNA. So point one is we get the cervical smear cells, we release and denature the nucleic acids and degrade all of the RNA. That's an important point. So we're just left with DNA, which is both viral DNA and host DNA. We then hybridize that DNA with an RNA probe that recognizes specific regions of the HPV genome associated with a high-risk type. We then capture these hybrids using an anti-DNA RNA hybrid antibody. So this is this antibody here. Now this is an antibody that just recognizes the shape of the backbone of any DNA RNA hybrid, so it's sequence independent. And we use this antibody to capture these hybrids onto a well of a multi-well plate. It's like an ELISA plate. Then the next stage is to then detect how much DNA RNA hybrids have been captured using a detection antibody. So this is the same antibody as here, only it's labelled with an enzyme which will then convert a substrate into either a coloured product or, in this case, a chemiluminescent signal which will give off light. So the key point is, if viral DNA is present, the RNA probe will bind to it. That will be captured onto the well by a anti-DNA RNA hybrid antibody. That will be quantified by the same antibody, which is added later, but that's got a label on it. That label is an enzyme that will convert a substrate into a chemiluminescent signal and light is given off. So you can have a 96 well plate system and the amount of light given off from each well is related to how much high risk viral DNA there is in that sample. So this is pretty much the same points explained, but it's sort of going through each step. So samples lysed, DNA is denatured, RNA is degraded. That's not mentioned, but it is. We mix it with the RNA probes. We get the anti-DNA, I should say RNA hybrid, antibodies immobilized onto the plate. So we have the sample mixed together, added to this antibody that's immobilized onto the plate, and then we quantify it with this antibody. So it's a fairly simple system. There's no nucleic acid amplification. There's no opportunity for contamination of tomorrow's experiment. This is just some details on the detection system. So it's an alkaline phosphatase cleavage of this substrate that results in an end product of these two, which you don't need to know what they are, but the end product is a photon of light given off, and that's what we detect. So it's not a nucleic acid amplification technique, and that's crucial because some organisms are only allowed to be tested by nucleic acid amplification techniques. So there's a hybrid capture test for chlamydia and gonorrhea, but it can't be used in the UK because the guidance for chlamydia and gonorrhea testing is it must be a nucleic acid amplification test. Hybrid capture is not. It's not sensitive enough. So chlamydia and gonorrhea are detected using a strand displacement assay, which is a nucleic acid amplification test that could contaminate future experiments if those products got out into the lab, whereas for hybrid capture there is no chance of that because there is no generation of high copy number DNA. There is a variation of this, which is hybrid capture, where you have your hybrid capture antibody detecting the DNA RNA hybrids, and then you can detect elsewhere on the DNA with an RNA specific probe that's specific to certain DNA sequences, and this will probably be called hybrid capture 3 when it comes out. Now the other way that this hybrid capture can be used is it's called baiting, where we can use this RNA probe to pull down certain DNA species, and then rather than just detecting the amount of them, we could elute off all of this DNA and RNA and put it through a DNA sequencer and then sequence everything there. So we're using this RNA probe as a bait to pull down just the viral DNAs, and then we can just sequence those DNAs, and you might have got a few thousand bits of viral DNA, which is going to be very easy to sequence using next generation sequencing technology. So that is a strategy that's been employed by some labs. So the hybrid capture 2 is actually very insensitive because it's not a nucleic acid amplification technique. So PCR can detect down to a handful of viral copies. Hybrid capture is about 5,000, and the makers of the system insist that their limit of detection just happens to correlate with the chance of the infection being important. So what they're saying is above 5,000 viral copies, it means it's a high risk of the HPV causing significant disease. If you've got less than 5,000 copies, which just happens to be their cutoff, it's probably a clinically irrelevant infection, but PCR would give a false positive there. So this shows how lesions can change from normal to cancer, and the normal cervix will go on to be a HPV infected cervix, which can become a precancerous lesion. But notice these arrows go backwards. A precancerous lesion can become normal, and we get cytological changes around here. Now if we get a persistent infection, and this has to be much more than 1 year, that's where we start to get the high grade changes. So it has to be a long-term infection, typically associated with stable integration into the genome. So if you've got a low level infection, maybe you come back in 6 months and see if it's still infected, in which case if it's clear, no need for further treatment. And that's where the HPV test is very useful, because many infections self-resolve. So you have an algorithm like this where you do routine cytology, see some abnormal cells. If they are high risk for HPV, perform colposcopy, so that's close examination of the cervix and maybe biopsy or treatment. If they're negative for high risk HPV, maybe don't even do any treatment, call them back in 6 months' time, repeat the pap smear, repeat the HPV test. If they're still positive, or if they are now positive for high risk type, colposcopy and treatment. If they're normal, go on to routine screening and come back in a few years. If they're negative for a HPV high risk type, go on to routine screening. So what is actually just starting to happen is, prior to the routine cytology, actually doing the HPV test first, and then basically only bothering to look at cytology samples that have got the virus in them, both low and high, but only bothering to look for cytological changes in those that have got virus in them. So in summary, the HPV hybrid capture test or the PCR based tests are going to limit the number of patients who are brought back in for invasive treatments that are not necessary. And they're not necessary because those lesions are never going to develop into cancer. They may well be a cervical wart and so on, but they're not cancer. So based on the viral type, we can reduce the amount of unnecessary treatment to patients and return low risk patients with a low risk viral type back to routine screening. So hybrid capture is non-PCR based. It's one of the few techniques that we're studying that is not a DNA amplification technique. What we don't yet know is whether hybrid capture is better than PCR. It's certainly easier and there's less contamination, but we don't actually know what is the best method. Within the whole HPV screening story, vaccinations are likely to play a key factor, because some of these types, all young females are being vaccinated against the major high risk types. So we'd hope that would result in fewer positives in the future.